I would set it in the morning, and then by like 3 hours later, it's 10 minutes slow again. It's really inconvenient.
So you're on multiple watches. That's what you want to say.
Yeah. I mean, I had a backup watch. Yeah.
You've got a thing on your foot that tells you the time.
Yeah. Well. Yeah. Yeah. So that's what I was doing yesterday. I kept being like, but I just like my wrist feels naked. Even if it's not functional.
Good sweat, man. Summer's coming.
Yeah.
You can just elegantly wipe your
Yeah.
forehead.
Yeah. Very functional.
Were the ESJ talks recorded or live streamed?
I think our session was live streamed, maybe not recorded.
I should have asked you this question before the session.
Possibly. I think some of the general, like oral presentations or whatever, recorded, but I think like 2/3 of them, yeah. Actually, I think they are recorded because they're like stored somewhere, and you can access them for up to a month after the conference. So actually, I think it is available. I would not know how to access that. You can ask Takahiro when we see next week. I know how to do that. Cool. I guess we'll get started. Without unplugging you. Okay. Yep. Hi, everyone. Thank you very much for coming along. We've got a bonus talk today. Fred's a bonus. He was not part of the original schedule, and then I realized he was going to be here, and I bullied him into giving us an extra talk. So thanks for that, Fred. Yeah, it's my absolute pleasure to introduce Fredric Windsor, who's here from Swansea. That's not true. Cardiff University. I'm getting confused now. There's two of you from Wales. Cardiff University. So he's a lecturer in the School of Biosciences. He runs the Freshwater Networks Lab. So he's going to be talking to us all about freshwater networks and particularly measuring the diversity of ecological interactions in the real world. So please, Fred, take it away.
I think that's on.
Yeah,
yeah. Fantastic. Thanks. Yes, so I'll get into talking a bit more about the work we do. It's kind of newly established lab at Cardiff. But yes, just to say, obviously, I appreciate that flowers and butterflies aren't fresh waters, and I'll get into a bit of that because we've had a little bit of a name change. So today I'm going to be talking about how can we measure the diversity of interactions that we see in the real world, and mainly asking the question if it's possible to, and whether we need to make use of or develop new methods to do so. So yeah, it is the Freshwater Networks Lab, but since I conceived the idea, I've realized I am interested in freshwaters. I'm a freshwater ecologist by training, but actually I'm also interested in pretty much every other system. So now I refer to it as Fresh Net Lab, a really clever way of kind of partially turning it into an acronym or funnel, where I cross out the fresh water. So it's Fresh Networks Lab. So it's all about fresh ideas, sometimes in fresh water, sometimes not in fresh water. But I just wanted to say today I'm going to be talking about mainly about this kind of one of the themes of the group, which is looking at developing mechanistic understanding of ecological interactions. But actually, I'm quite a generalist, and I like looking at anything really that interests me. And so we have a whole range of other projects running in the lab around long-term trends in different communities, linking in things like biomechanics into networks, a lot of networks, but also a lot of other interesting ideas. And I'll leave that on there for a little bit to see if there's anything that takes your fancy. But this is just one aspect of the work we kind of do in the lab. So I wanted to try and take everyone with me on this talk. It kind of dots around, and it's very much a talk of kind of new ideas and new areas that we're exploring in the group. A lot of unpublished stuff in here. So I thought I needed to provide some kind of background, get us all on the same page before we kind of delve into some of these different aspects of the work that the group are doing. And key is understanding what I mean by ecological interactions and ecological networks, because it's incredibly it's an incredibly diverse field. Lots of different people define them in lots of different ways. Lots of people work on lots of different aspects of interactions, both in space and time, between within species, between species, for example. So I just wanted to cover a little bit of ground on what I mean, at least for the purposes of this talk, rather than what is meant by them as general concepts. So a lot of the work we do is focused on these freshwater networks and the interactions that take place within them, and particularly interested in looking beyond where we're currently looking in terms of interactions. So a lot of work to date is focused on clearly critically important direct interactions, things like predator-prey interactions, predator-prey in the context of herbivory, so things eating plants, competition, parasitism, but less so potentially on these kind of less direct or indirect interactions where you have things like commensalisms, which take place through this indirect mediator of some sort of abiotic environment. So here, disturbance of the bed by a spawning salmonid and its impact on an invertebrate. And I wrote this kind of perspectives piece to try and summarize that way of thinking and start, especially in freshwaters, thinking about a wider diversity of interactions. It's certainly something potentially, especially in stream ecology, that we've not been particularly good at, is appreciating things that aren't related to food webs or trophic interactions. And I think those positive interactions, mutualisms, but also kind of commensalisms, have the potential to be really important, but we just simply don't know enough about them. So that's a really broad description of different kinds of interactions, and I'll focus in on a few of these key examples as we go through the rest of the talk. But we also look at networks, and again, networks are quite broad, and there are different types of networks. We have individual networks between individuals of the same species, things like social interactions, behavioral networks, through to these kind of interspecific interaction networks, which are the mainstay of most of network ecology, where you're looking at how different species interact with one another and potentially the structural or functional implications of those interactions. And then more recently, a whole raft of work has been done on incorporating space under the guise or using information and theory from things like metapopulation and meta-community work, taking a kind of meta-network approach, although that has kind of different implications in the field of network ecology, certainly looking at space and including space in some kind of meaningful way. Again, some of the work will focus in on some of these aspects, so just so you're aware of these kind of different levels, different scales, different types of interactions. So a key question I always ask myself, I'm a community ecologist by training, is do we need networks? Do we need to understand interactions? They're a pain to try and detect. They're often very hard to analyze. The best thing you can do if you think you need to look at interactions or networks is ask, do we actually need to, or can we use something simpler to understand what we're trying to look at? I would argue that in a lot of cases, we can use something simpler, but in some cases we can't. And in particular, they're important if we want to look at those links between structure and function. Quite often those interactions convey some sort of ecological process which converts our community or ecosystem structure into some kind of functional output. And by proxy of that, kind of give us a mechanistic understanding that we might not get otherwise. And also critically, and I won't talk much about this today, but we have been doing a lot of work, and lots of people have been doing a lot of work on appraising indirect effects. And you can do that quite neatly and quite elegantly through using interaction networks, because you look at those linkages between species or individuals and look at indirect effects. So this is kind of the key for today, but don't worry, we're not going to focus on all of those aspects. I realize I switched them around in the second slide. That's very confusing. Let's just look at this. Is we're going to focus on some of the key gaps that I personally think are there, obviously. So one of them is expanding our coverage of interactions. So again, the majority of work is focused on a specific suite of interactions. Is it possible to look wider using new methods or different methods? Information on drivers or mediators of interaction. So again, understanding how and why interactions change in different contexts, both biotic contexts and abiotic contexts. And that's to do with the kind of understanding those mechanisms. And then also quantifying interactions. So looking at how strong interactions are between those nodes, between whether it's individuals or species, is an interesting space to look at. And I'll pose some questions in that section, as well as what do we mean by strength of interaction and how do we quantify it. But this is kind of, network ecologists think of a quantified network as like the gold standard, the maximum amount of information you can gain on a kind of ecosystem. So there's been a push towards trying to quantify these interaction strengths, and kind of ask some questions and pose some methods for doing so in a consistent way across different interaction types. Or at least I'll try and do that. It turns out it's incredibly challenging, hence why no one's done it yet, or done it much. So first thing to start with is monitoring the diversity, or expanding our focus into different interaction types. So again, like I say, I'm always quite self-reflective and self-critical. So do we need to look at other types of interactions? And again, obviously, I wouldn't be doing this if I didn't think we did, to some degree. So the key thing here is we're very good at looking at certain types of interactions, and we have quite well-established methods for looking at, for example, pollination or predation. Things like stable isotopes, dietary analyses, dietary metabarcoding are all very well-used methods for looking at trophic interactions. But what we're not so good at is looking at non-trophic interactions, and specifically kind of those indirect non-trophic interactions. And that's critical, right? I was saying about indirect interactions potentially being very important, and it's important that we understand them. So if we're not looking at all of the types of interactions, or not looking at those indirect ones, we're missing out on quite a large kind of gap, quite a large area of our interaction networks. So that leads to the question of, well, what should we be looking at, Fred, if you're saying we're not looking at the right things? And that's a big question. There are lots of different things we could look at, and therein lies a challenge. I talked about spatial dynamics, which is a very good area of research, very interesting area of research, looking at measuring interactions and links in space. And that's a bit more abstract, in the sense you might be linking a species to different habitats in the sense it visits those different habitats. So we're not going to focus on that today. You could also think about linking all of these different types of interactions we already have data for together into some sort of massively complex multi-interaction network. And again, that's kind of a bit of a minefield, so we're not going to look at that today. But what we are going to have a quick run through is a recent project one of my undergrad students has been working on, around looking at interest-specific interactions, taking a reasonably stepped back approach, quite a naive approach, but I'll talk you through some of the things we've been thinking about, and certainly happy to have feedback and input on this. So this work has been looking at stream systems. So this is a freshwater project across 4 different upland stream systems along a gradient of what we will term harshness, but it's essentially a pH gradient, so from acid through to circumneutral. And looking at 2 functional feeding guilds. So in macroinvertebrate communities, we typically group those invertebrates into these different functional feeding guilds. So grazers, if they're scraping biofilms off the surface of rocks, or shredders, for example, here is shredding up leaf litter that falls into those systems, or general detritus in those systems. Looking at 2 different groups, which are relatively different. One predominantly feeds on inputs, allochthonous material, and the other feeds on typically autochthonous, which is produced in stream from diatoms, various other kind of microbial taxa. And using stable isotopes, and the crux of this is we've just got a fancy new stable isotope analyzer at Cardiff that can do nanomolar levels of 15N and 13C. So we've got the ability now to look at levels of these heavy isotopes in individual organisms, but even in the actual sub-tissues or like organs of these invertebrates, depending on their size. So this is what this all fell out of. And this is my excuse for some probably like theoretically very naive next ramblings around how we can try and quantify competition from some of these data. So caveat. So we've been thinking about how we might do this, making use of this fancy new bit of kit where we can analyze levels of these stable isotopes and say something about their diet, their dietary niche, by amalgamating this kind of, or not amalgamating, but by looking at the spread of these individual diets. And so we've been thinking, can we look at intra and interspecific competition? So it's not per se a kind of network challenge, it's an interaction challenge. But we've been thinking about different methods, and so this is where the naivety comes in. So we can generate, and what we have been generating, and not generated quickly enough, hence why it's a PowerPoint diagram rather than an actual scatter plot, is looking at, for say here, 2 different species, what the overall kind of niche breadth is based on stable isotopes and their kind of the delta 15 and delta 13C, what their niche breadth is, and whether we can say something about the levels of inter and intraspecific competition that might be going on in those systems. And we've been using niche variation hypothesis to say something about how this niche breadth might convey or have some impact on the likelihood of intraspecific competition, which is somewhat challenging, seeing as that niche breadth is always also dictated by interspecific competition as well. So we're still working through these methods, but our idea is to try and get some quantification of intraspecific competition to tie into our understanding of interspecific competition within some very kind of simplistic interaction models. I won't talk too much more about that because that's quite early stage, but what we've also been doing is thinking about, well, you know, usually we do collect multiple samples for our interspecific stable isotope analyses, but we typically represent them as a kind of mean value plus or minus some sort of error. And we have been doing analyses such as this, but can we do it at a higher resolution, or can we get a better estimate of kind of niche overlap as an indicator of potential interspecific competition? Bearing in mind the caveat that these species are already occurring, and therefore, you know, competition is acting to restrict these niches or impact these niches. Can we use this as some sort of measure of competition? And we've been developing some simple distance-based or niche overlap models based on some of the work, and some very nice packages in R. So we're trying to link in, as well as we'll have kind of trophic structure and interactions from Bayesian approximations. We'll also hopefully have some sort of indication of relative competition in a slightly more quantified way than we would have had previously. So kind of out the back of that, we have some general hypotheses, which we're in the process of testing around kind of intraspecific versus interspecific competition under different levels of resource limitation. Those different streams have different resource availabilities to those different groups. So the circumneutral streams have much higher levels of autochthonous production. So they have lots more grazers, but they also have a lot more resource in comparison, and less overall relative resource for the shredders. And the converse is the case for the acidic streams. So by doing some interstream comparisons based on what we've done, we're hoping to get towards some sort of idea of what is limiting or affecting these two processes. And we also want to look at whether those competitions between individuals within the same species change. So similar to actually the above question. And I suppose I really wanted to talk about next steps as a result of this. And we've seen, based on some initial analyses on some very limited data, that obviously they're both there. They're relatively comparable from what we can see, but at the moment, I'm loath to say it's due to the ecology. I think it's probably just due to the kind of relative roughness of our methods, or some sort of lack of understanding on my behalf, I would say. But key here is about verifying the reality of these interactions. And I'll talk a bit later on about the use of emerging technologies in this area. And hopefully that'll be quite interesting. So that was kind of the first section. We've got two more, both of which are a lot quicker, so don't worry. They kind of link onto one another too, so hopefully it makes a logical flow. So the next kind of sub area is talking about okay, that's interesting. Different types of interactions. We can potentially expand to look at other forms of interactions. But what's actually driving or influencing those interactions and their strength in these systems? And this is where we've been developing as a group, and in particular, one of my grad students, who's an electrical engineer by training, which is fantastic. I would highly encourage you to look at engineers as potential students if you're not already doing so, because they bring a whole different skill set to the game. And we've been looking at whether we can use, it started with kind of audio and, started with videos and images, which was my idea, and it's merged into kind of audio visual techniques to identify interactions, but also understand what's driving them. So we've been using audio moths, kind of initially, but now Raspberry Pi-based camera and audio recording setups to try and understand interactions, but also understand the context within which they sit. So this is a very nice drawing, not by myself, unfortunately, but by my grad student guy, of a kind of mock setup for looking at pollinators in a kind of experimental field system. So you have these different sets of cameras and their processors, which are those boxes. You also have a kind of weather station set up as well. And certainly now with these new bits of tech, you can set up weather stations for a couple of £100, which is pretty impressive. And what we're doing at the moment, and we're just setting up our first kind of field trial, experimental trial for some of these tech. We've been testing them in the field, is to look at whether we can both film and also record audio for other purposes I'll talk about in a minute, to look at detecting interactions and their strengths. And it's really good because there are a load of these green benefits of doing this. There are also some reds, which are slightly challenging. But using these video approaches, we can go beyond our previous observational studies, especially for things like plant-pollinator interactions, where it's a case of being out there and physically observing those interactions, recording them, monitoring maybe how long they go on for. But that's as far as we go. With these video methods, we can get non-target species, we can potentially get better levels of ID, or at least we can double check those IDs, whereas in the field, that's very challenging. We can also operate for these systems for longer than we could do based on visual observations. And we can also, which is where we've been focusing a lot of our recent work, is trying to integrate different scales of movement. So from micro to macro. So you understand the how and the why of movement and interaction, as well as just the actual interaction itself. So in particular, we've been looking at using some different secondary data sources, as well as thinking about it for our studies, to look at things like time delays in interactions, where you might get kind of these negative co-occurrences, but offset by a period of time. So you can imagine a strong competitor visits a plant or a flower for some period after, based on either the pheromones or the key cues it's emitting. You might not get those other competitors visiting that flower. So you have this kind of time delay, this time lag that prevents an interaction that, again, you just wouldn't detect using standard methods. And in particular, for some camera trap work we've been doing, not on plants and pollinators, but just on urban vertebrates, is looking at this idea around this landscape of fear, which I think is quite interesting. So there's been a lot on energy dynamics around landscapes of fear between prey and predators, but also thinking about how that landscape of fear might influence the interactions. So again, you can imagine if there are food resources for like an omnivorous vertebrate in an area, the decision as to what food it should make use of might be influenced by the presence or absence of a predator. Some foods might be more nutritionally beneficial, but harder to access. Some foods might be less nutritionally beneficial, but easier to access. So actually, its choice of interactions in that instance is dictated by this exogenous input from a predator, whether it's visual, auditory cues, that you just, again, simply wouldn't detect using standard methods. And that's where the audio moths come in. So I've been doing work on detecting predators in the external auditory environment, and then looking at interactions between organisms in the visual environment, and looking at whether there's any link between those two. And kind of finally along this thread, we've also been looking not only at kind of how biotic factors influence interactions, we've been looking at how the environment influences interactions. And this stemmed from some work with a colleague, led by a colleague at Newcastle, where we found quite significant patterns in trophic interactions that we detected using DNA metabarcoding of gut contents in spiders, and showed that actually this signal was not just random, it was related to weather and prey availability. And it showed that spiders are limited in their diet in different times of the year based on the weather and the availability of different prey types. Some of which is kind of confounded by the fact that prey are either available under those certain weather conditions, or it's the time of year when those prey are more abundant. But there's certainly some influence, direct or indirect, of temperature. So again, similarly, we've been, as you saw from that diagram, we've been setting up our weather stations to monitor these critical variables, especially for ectotherms, things like temperature and humidity, or for our insects, for our pollinating insects, temperature and humidity, to try and understand again whether certain interactions take place under certain weather conditions. And certainly, obviously, the frequency of interaction might be affected, especially in ectotherms, all with the aim of trying to look at what are the potential mechanisms. And then this is a bit of a kind of tack on that I thought I didn't want to not include this fantastically kind of blinding graphic that Jordan put together, Jordan Cuff. It's this idea of nutrients, and I kind of talked about it around the landscapes of fear, but I think it's got a wider importance in terms of structuring these interaction networks, these trophic interaction networks. For vertebrates, for example, a lot of work's been done on nutrient limitation, for example, and selective foraging based on nutrient requirements. Less work's been done on invertebrates, but certainly enough to suggest that actually these might be key drivers of our interactions. And certainly we're trying to factor in nutrients and cues for nutritional quality that predators can detect into looking at our network structures. So final topic before a conclusion that somehow has to synthesize those three is around quantifying interaction. So I said this is kind of like well, it's not heavily contested, but it's seen as a kind of gold standard. If you can quantify an interaction in a meaningful way, that gives you kind of maximum information on those interactions and gives you maximum value, bang for your buck for your network. But it's very hard to do, and it's often done very badly. Or we use proxies of interaction strength, which are more or less related to what it is we're trying to look at in terms of an interaction. And I think the key thing when I've been thinking about this work is actually what should we be measuring to look at interaction strength. So classically, we've been thinking about the interaction itself. So for plants and pollinators, we've been thinking about the time that a pollinator spends on a plant as a proxy for the amount of pollen it might be picking up, again, for a proxy of the amount of pollen it might then deposit in a receptive flower that leads to pollination. And there are lots of other examples across different types of interactions. And that's good. You're focusing on the interaction. Obviously, that's the thing we're measuring. But does it actually denote a interaction, if you see what I mean? That doesn't necessarily translate to pollination, the amount of time spent. So it's an open question, but a question should be asked nonetheless is, should we be measuring those interactions, or should we actually be looking at the functional implications of those interactions as a measure of the strength of the interaction? It's somewhat circular and somewhat linked, but it's an interesting way of thinking about either some new methods or some new ways of thinking around this. So we've been doing a few different things. One of our ideas is on plants and pollinators and looking at using our video setups to look at interactions, frequency, time, things like that, and link it through to the seed set of the plants. And we're doing that this summer. But another one, again, using some secondary data before we could generate our own, is to look at the kind of behavioral implications of interactions and look at how behavior changes before and after an interaction, again, to give some indication of strength. So that's a little abstract. So I'll talk through what I mean by that. So what we've been doing is looking at some experimental data, as you see, very fuzzy, on Drosophila, on Drosophila melanogaster, and looking at some what are termed aggressive interactions. And this study in the Journal of Experimental Biology, I'll just play it one more time so you can see them, focused on kind of social hierarchies in Drosophila, and the kind of dominance, competitive dominance or aggressive dominance of certain individuals. I don't work on Drosophila, but the key thing for me is that it's quite interesting is that there's a labeled video data set here with different key interactions, things like tussling, which is a kind of aggressive interaction, wing flicking, which is a kind of warning interaction, and lots of other different types of interactions before or in and around those, such that we can look at whether those, what we term competitive tussle interactions, influence the behavior, and look at before versus after to see what the potential functional implications of those interactions might be. Oh, there we go. So again, this is Guy, one of my grad students, has been working on looking at these are, we haven't really come up with a name for it yet. Someone's probably already done it, so I'll just look through the literature and find what it's called. But these are the principal components of a load of different behaviors that we've codified ourselves. So it's all around angle of movement, speed of movement, proximity to other individuals. But essentially, this is kind of like I suppose, a behavioral trait space. I suppose is probably the simplest way of defining it. So here, where we have these, and this is just density, right? So yellow's high, blue's low. Here we have, the majority of our individuals are kind of occupying this area of trait space, which is related to a certain class of interactions, group of interactions. And then we have another peak over here. So this is in our before our tussle events. And we've been doing this across all different kind of windows of time. So short durations before, long durations before, short, long after. This is just a snapshot. We then have this tussle interaction, which again is characterized different scales. Anyway, characterized by a slightly different set of interactions. And then afterwards we have, that's the difference, sorry. And then afterwards we have this general kind of malaise of no clear peaks. And what we see when we compare before and after is that we get these differences. So we lose those peaks essentially, which is obvious if you look at that. This is all kind of very non-specific. We don't really know what the functional implications of these interactions in this instance are, but you can see how that way of thinking might lend itself to something further. And certainly we're asking ourselves, can we use techniques like this to kind of quantify interaction strength in a more functional way? I suppose that's the key take home. So just to conclude, so there's a bit of time for any questions that anyone might have. I think the key for me is that actually measuring interactions and I suppose collecting any biological data is not a trivial task. There's lots of different ways to do it, and they're all flavors of right or wrong, depending on whether you're kind of glass half full or glass half empty. I think emerging technologies have promise, but they also have key limitations. One of the things I didn't talk about is obviously when you're videoing, you have a defined focal distance. You have you're spatially very constrained. And that's a trade-off between resolution and overall coverage and things like that. So, they're not a panacea, they're not an answer, they're not an alternative, they're kind of a supplement in terms of the methods we're currently using. And I think key for the future maybe is less so thinking about new ways or different ways of doing things, but it's more about trying to integrate some of the data that we do have, or integrate data collected from different methods such that it kind of makes sense and is rational. And certainly that's when you start to combine data on different interactions that have been measured in very different ways, you run into massive problems around biasing whatever analyses you subsequently do on it towards different interactions. because they were measured in slightly different ways. And we've been doing quite a bit of work on that, but it's a bit drier, it's slightly less exciting. So I thought I'd leave that one out today. But certainly we've kind of proposed some methods which maybe we don't. Historically, what you do is just binarize the network. So you just have the interactions present or absent for like competition or predation if you're having both of them. Obviously, by doing that, if you've tried to measure interaction strength, you've lost all those data. What we've tried to do is use some other methods to lose less data when integrating those data sets. But yeah, happy to have a chat about that afterwards. But with that, I'll conclude. So that's what we hope to get towards. Whether it's useful or not is the question we should always ask. But certainly looks nice. So yeah, I'll leave it there just for any questions. I think that's about right for time.
Thanks, Fred. That was really nice and very different from everything I kind of know you for. So yeah, really cool. Plenty of time for questions if anybody has anything. But yeah, please use the mic because there are people on Zoom.
Really interesting talk. If you can go back to the conclusion slide, I've been thinking about this a lot as well. And it reminds me of the Lafferty paper from a while back that was like, parasites dominate food web links. What are the implications of this? And I remember reading that and thinking like, yeah, it's because there's a bunch of parasite species, but parasites in terms of what that link costs the host relative to trophic outcomes that are like predation directly are very different. And that has really big implications. So I was wondering, and you've probably thought a lot about this, how do you do the thing? Like, how do you get at that?
Yeah, I think it's a big challenge. Excuse me, you're right. Yeah, there's this continual battle between kind of protagonists saying, one interaction is more important than another. I think it's key in networks is, hey, you haven't looked at competition. Look, it's key. Well, yeah, we know that all interactions have some positive or negative influence. And I think, that's where trying to get them on the same operating level is important, but very challenging. That's something, I'm not an experimental ecologist or biologist, but I do wonder whether there's some, and lots of people do this work. And I wonder whether that's where it's important for us to kind of work collaboratively on some of these questions, because I think the functional implications of interactions are really hard to get at in a field setting, because just like everything in the field, it's tied into a load of other things that vary. The trouble with things like parasites is there are so many different parasites and so many different levels of parasite burden that trying to get a meaningful answer, even through using experimental, looking at condition, fish, for example. Vittoria, one of my students, looks at parasite fish dynamics in catchments in Brazil. There are so many different species. They also co-occur, don't co-occur, like trying to actually even get some, per fish estimate of, I don't know, impact, I suppose. And then that will be at the individual level. How that scales to like population level, again, tied into communities, is really challenging. But I think it's a thinker. This has like opened a whole can of worms for me. I was just started off thinking, hey, can we use some of these cool bits of tech to detect interactions? And then it's led down this whole thing around like, well, what are we detecting? How important is it? Is that the right measure? And I think it's something, there's no right answer, but I think it's really interesting to think about. But I don't actually know how we do a lot of this. So maybe I should stop before it's too late.
Other questions?
Thanks very much, Fred. Really interesting. Intra-specific competition and inter-specific kind of inferred from the delta 13C and delta 15, is it?
Yeah.
I think you mentioned that you're going to try and validate that. I was thinking about that before you mentioned that, and how maybe one could look at trait similarities among the individuals or feeding apparatus similarities,
Yeah.
without then having to go to maybe more sophisticated types of approaches. So were you thinking about that?
Yeah, that's a very good point. I was having some really good chat. So sort of macroinvertebrate trait analyses is pretty a relative, well, I'm going to say this with big quotation marks, relatively limited. I mean, for a lot of our trait analyses, we don't anymore, but certainly we're using these big trait databases, which are incredibly coarse, I would suggest. But yeah, that's a great point. In terms of, I think if we were to do that, measuring those traits on the individuals in those streams and getting some sort of trait distribution, some probability density function or something to actually just look at overlap in those traits might just be a way simpler way of doing it.
So about the individuals that
we have some individuals, but that's a good point. Yeah, because with these methods, actually we could take the heads off, for example, in this instance where we're talking about mouthpart morphology. That will make no difference to whether we'll be able to get a detectable measure. So you could take the heads off, so you can keep those for those individuals and look at and pair things like those traits of those individuals, or measure them before and then process them. But you could pair individual traits with individual kind of, individual dietary contributions. So yeah, no, that's a good point. I hadn't actually thought of that. This is kind of like a preliminary project. And I said I was a generalist at the start, and it's just, well, I say about my naive understanding of interest-specific competition, but it kind of just gets me thinking about these things. But yeah, I think that's a great suggestion. And that's the power of these, is actually we're not aggregating in any way. We can tie those signatures to individual organisms and look at their traits too. Yeah. And we're currently working with the technician to see how small we can go. So we're hoping we can actually trace, it wouldn't be the same individual, but look across growth trajectories or across different life stages, different ages of these invertebrates, and look at those, how that changes over time. And that could tie absolutely into things like mouth morphology and relative size. So yeah, that's a great point.
Dave?
Thank you. So sort of, my personal bane as a community ecologist is the difficulty in using some of these correlation network approaches to infer species interactions on one hand, and then on the other hand using experimental results to say anything predictive about co-occurrences. And so there seems to be some sort of fundamental mismatch that we're just not able to deal with. And I guess, what's your opinion on that? Do you see it as something we'll never get around, or do you think kind of merging all of these different technologies is going to get us there? I don't know. Are you optimistic about kind of solving this, major issue?
Yes, I am optimistic. No, I think that's where we kind of landed with these video methods. I was thinking, what do a lot of people do, or what have a lot of people started doing in the lab when they're looking at running behavioral assays, in terms of looking at some of these small scale behaviors and maybe trying to upscale them. And a lot of people have switched to using this rather than direct observation. Especially as our kind of computing powers increase, we've been able to run some of the algorithms we use pretty easily. So I think I see kind of the techniques we're trialing for these, on these kind of plot scales, there's some sort of intermediate between the two, or some sort of bridge. Again, I don't think it addresses that key problem. And we have been doing work trying to infer interactions from long-term data sets, for example, and co-occurrence across long-term data sets. And it's completely unsatisfying and throws out a whole load of kind of quite spurious results, at least the way we've been doing it. Maybe someone else has done it better, but it's quite challenging. So I am, maybe optimistic. I'm certainly not shy of trying to address it, but whether we'll actually get there or not, I don't know. You kind of just move into a different area and then realize it doesn't work. But I think maybe these kind of merging methods offer some way of kind of, especially that's what actually, I suppose, the thing that we've been doing as well is not just using different methods for different interactions, but using different methods that have some overlap. such that you can look at what interactions they detect for a shared interaction that they can both detect, and using that to say something about the wider spread of interaction. So that was not particularly elegantly put, but I suppose what it's saying is, like, at least for those shared interactions, you know what they do and don't detect, and you can then have a bit more confidence when you're kind of merging or integrating those data sets. But
like a vague sort of Bayesian, working across all available evidence might be a better solution.
Yeah, that's true. Yeah, and I think, I can't remember, I want to say it's really recent, but my ability to read papers over the past year has been somewhat limited, but I think it's relatively recent suggestion of, Bayesian approaches for integrating data. I think that was community. Maybe it was even you, Dave. I don't know. But there was something that came out recently, and I did think, oh, okay, yeah, maybe that is a good shout. So yeah, absolutely.
And I'm just going to sneak a final quick question in.
Sure.
So your stuff about the landscape of fear, I think, is really interesting, especially using acoustics. I didn't know. Is that maybe a leftover from the Silent Cities project? I don't know. But I had no idea you were doing this. But I mean, I've grandiosely been trying to write a paper called The Soundscape of Fear for a few years now. But I wondered if you were thinking about playback experiments. So you were talking about monitoring what predators are out there, and then using these multimodal techniques to think about interactions. You could also just play predators at them maybe and see if that makes a difference.
It's a very good point. Yeah, so this is part of actually that soundscape of fear. Most of my work is on invertebrates typically. We got some money from the university as part of their kind of sustainability push to try and do some biodiversity. Well, I think they want biodiversity accounting, but that's not really what I want. But anyway, to monitor the biodiversity present on campus at Cardiff. And so they said, right, what are the best ways of monitoring biodiversity? And I just kind of threw out, well, we can do this. You already have these cameras doing this. Like, we can just merge them together. So that's where, excuse me, the ecoacoustics came from. Because I thought, well, one thing that we have quite an active kind of wildlife society that collects sightings on birds. And obviously you can use NBN Atlas and things like that to collect, kind of ad hoc observations of different species across campus. But those ecoacoustic, you say Silent Cities, things like that, and your work as well, Sam, on detecting different species of birds. It's quite a unique and quite an interesting way of looking at that group. So kind of threw those in, but it's also pairing those cameras and audio recorders just kind of, allows you to potentially do that. So it's very much the reason you haven't heard about it is because it's probably about 3 months old. So but no, absolutely. I think, it's interesting you're working on that already. I knew someone, as with all of my ideas, someone else has done it. And in network ecology, it was usually in the 80s or 90s, so even more depressing. But great, thank you.
Awesome. Well, thank you very much, Fred. Cheers.
Yes.
We're going to take a quick 5-minute break before Steve's talk, and then we'll